Okay, so good day to all of you. So today we are going to start the 2 days training program on quantitative data analysis. Yeah. So before starting, I would like to announce some of the things. First of all, that regarding the certificate, who have attended the whole course with more than 80% attendance, they will receive the certificate. This is one thing. Second thing is during the whole session, if you have any questions, you can ask me. It's better, no issues, but it's better to type in the chat, or maybe you can ask during the session. And the third thing is that all the theoretical part, the presentations and all the code in R, all will be available in Google Drive. I will share the link tomorrow after the end of the session. So you will receive all the materials related to the whole course during the time. This is one thing. And another thing is that the meeting is fully recorded. So recorded version is also available in the YouTube. So you can maybe 2 3 version. So we will distribute it after the during the with the certificate. So you will receive the recorded version also. And another thing also, I want to say thank you all to participated in this program. I hope that it will be helpful for you in your research, in your future career, or maybe in your higher study. So this is the program is basically uh, this is a program basically make things that can be basically available for. Okay, so it's available for means any kind of science. It's not restricted in biological science or any kind of science who are basically doing on their research in and then we are facing a lot of problem during the data analysis. So that is I try to make it as simple as it possible. So basically, this is the basic course. We are not going to into the deep of the analysis, but yes, somehow I will tell you about the introduction of the statistics and also some of the basic test and over regression correlation is very essential for a scientific research. So I will this is divided into two part. One is the theoretical part. First I will explain the theory in maybe 1 hour, and then next 1 hour we will do the practical session running the R programming. So here everything is data is available, code is also available, and the presentation also will be available. And and one more thing, I just want to share you one thing. Just a second. Uh, Window, yes. So the program is basically free. You don't have to pay as you register. Uh, yes. So the program is free, but if you are available for make it just to discuss this. Yes, it's free, but if you are willing to uh, donate some money, you can. So here is the link is available, and you can basically donate, and that whole money will goes for the for the needy children and their educations. So this is one of the thing I can just make it full screen. Okay, so it is available for I don't know, it is international, you can pay. So there is a two payment version. I have received maximum participant from India and then Oman. So almost 80% participant from these two country. So I have keep these two link, you can save. I can also share you in the chat box. So this is the two link, you can pay, you can donate as much as you can. There is a no limit, but the whole money will goes for the children educations. Actually the thing is that I believe the education must not have any boundaries, must not have any limit, and don't have any barrier. So I always try to give as much as possible from my side to donate. There is no organization, no party, no one involved. I directly conduct those kind of program to donate the needy children. Okay, so can you please mute your. Yes. So this is the link. You can donate as much as you can. Okay. So we'll start from the. So we'll start from the first program, that is the I will introduce the topic, that the data science. Then we'll start the one by one test, starting from the t-test ANOVA. Today we'll finish, and tomorrow we'll finish the regression, correlations, and the chi-square test. Okay. So if at the beginning, if you have any queries or questions to start it, maybe the program can be 2 hours, but if the time of if not we can finish, we can extend few more minutes or few more not hour, but yes, maybe 5:30 or maybe half an hour we can extend. It's depend upon the situations. So let's start the today's session. So before that, if you have any queries or questions, so you can write down in the chat box. So we'll see, and then we'll try to answer at the end. Okay. Open the second Windows. Yes. So is it visible to all of you?
Yes,
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 doctor.
bro.
This is
Yes, sir.
Okay. Okay. Good. Good. So as I told you that this is not a particular science. It is not a marine science. It is not a agriculture science. It is not a biological science. It is the science of all science. Okay. So here we all need to represent our data. We need to analyze our data, because each and every day during our higher study, during our research, we are collecting a lot of data. But after collecting the data, we don't know what to do with this data. So sometimes I find out that sometimes we have to go someone or find someone who can help us to analyze our data. This is very very common thing. Also in our time, we also don't know how to do this t-test. We know the t-test, the ANOVA, but we don't know how to do this thing. So we have to go someone who knows those things. So why not we are learning those basic statistical tools or basic analysis that we can do ourselves to find out the better way to develop our science of data. So here basically the data science is very common. It's everywhere. So if so why it is data science? So it is the most important thing. It is everywhere data is there. Okay. So if you go to any sector, it can be education sector, any sector, in transportation sector, telecommunication sector, corporate sector, business sector, anywhere data, a lot of data. In science there is also a lot of data. So but that data does make any sense to us when that data could properly analysis. Okay. So this is the most important thing. So because the data is only a number, that numbers, or sometime it have gives you some quantitative or qualitative data, but that does not make any sense until unless you do any analysis. So that's why we need some techniques or tools. To that techniques or tool basically will do the analysis, that will find out, tells you that yes, your data is can be, I will say one example later on. So that I told you that whatever your career it is, it's not belongs to any, it is science of all science. Okay, so data science is belongs to every science. So here the most important part of the data science is that there is a steps of data science, but ultimate goal of the data science is to take a decision. Okay, so we are collecting the data. After collecting the data, we are analyzing the data. After analysis, we will interpret the data, and then we come up in a solid conclusion. That conclusions make you tells you a decision, and that decision not only the scientific area, not only scientific research, that analysis is also very essential for the governmental sector, private sector, every sector. What is the growth of economical growth of a country, what is the employment, every sector they will, and even though we have seen the COVID analysis, okay, COVID-19. So all every day the graphs are coming, the analysis are coming. So without the analysis, we cannot say that this much a percentage COVID is distributed this area, this is the things. So until unless you are not analyzing the data, or you cannot reach in a valid conclusions or valid decision. And that decision not only the science, it's also the governmental decision, any company's decision, however the growth rate of the company is going high or low, what to do in the next step. So that's why they have to take a valid conclusion or decision will comes from the data analysis. So I told you that data does means only a number, or maybe qualitative or quantitative. Quantitative a number, qualitative good, bad, okay, yes, no, okay, very good, very bad, very good. Okay, so this kind of qualitative quantitative data we are receiving. But what is the from the data? What is basically comes from this? Come from these informations of this data. So suppose here we have a some data of a body weight of some of the persons. Okay, so from this data we can conclude a informations that five individual of this data range from 40 to 45 kg, and 15 individual from this data range 45 to 90. This is the one informations, but that information will also not tells you the whole things. So what is the analysis? We did a statistical analysis that mean weight of the whole populations is 54 kg, and median weight is 60 kg. Okay, so what is the mean median we'll discuss later on. Okay, so this is called a statistics. Okay, kind of a one example I can say that the a blood pressures of a populations, you measure 100%, 100 people their blood pressures, and then you find out that the high range of blood pressure of this much person, high low range of blood pressure of this much of person. Then you do a statistical analysis, then you find out that whether what is the population's blood pressures in population mean. Population mean is 120, 80 by 120, or 120 by 80. This is the population mean. Now you compare this population mean with your the data, with your data 1 to 100. Then you tell that yes, this population is suffering from a high blood pressure disease. Okay, whole population. It might be due to the job. Okay, due lot of pressure in this job. So this is government should take a decision that yes, the whole population is suffering from a high blood pressure because of the huge job pressures. So government should decisions at that job pressure or the work pressure should be reduced to relax the people. Okay, so this kind of analysis will come a valid conclusions or decision from every sector. That's why we need the data science. Now comes to the what is data. I told you it is the informations of number. Okay, whenever we are going, we are basically collecting the some number or value. Okay, some observation, some measurement, some counting, some response. Okay, we are going to counting the birds, how many birds are there. We are going to estimate that how many pH of the water. So any measurement, counting or observations, how many flies in that area, okay, or agriculture field, whatever it is. So this is called data. And what is statistics? It is basically science of collecting the data. First we have to collect the data, Then we have to organize the data. It is very essential that you have to organize your data in a proper way. So you are collecting a lot of data, but you don't know how to organize the data. That will makes you very difficult in terms of analysis. So first collecting, then second you have to organize your data proper way, then you have to do analysis. This is the third step. Okay, analysis means any kind of analysis, graphical presentations or statistical analysis that tells you that it is high or low, and then interpret the result. Okay, what happened in general science, what we are doing, we are plotting our data in a bar graph or in a line graph, and then we are telling this is big, this is better than this is big, this is less, this is more, this is low. Actually in statistics or in science, it does not make any sense whenever you are telling this is big, this is small, this is high, this is low. No, there is no sense of this telling of this kind of word in science. Okay, when it will be you do a proper test between these two variable, then you find out proper test, you get a result or statistical analysis, and then you can tell yes this is big, and with supporting the statistical test result, that is basically acceptable. Otherwise what we do just plotting some of the bar chart, and then we are telling this is high, this is low. Okay, actually this is not the way to proper presentations of your data. Okay, so if you are telling this is high and this is low, then you have to do a t-test between these two means. Okay, then you can tell a result, your t value and the probability value. Yes, then you can tell yes this is bigger than this one with statistical result. Okay, so this is the way how you can do a statistical test and interpret your result. Yes, this is high and this is low with proper statistical test. Then you can make a decision. Yes, this variety is good in compared to this one. So the farmer should choose this variety. Okay, for their economic benefit. Okay, so this is the way you have to do your analysis, and you have to interpret your result, and you have to make a decision. But there is we will talk about a different approach that is called statistical error. That is also one time happen. So if after statistical test I am telling that this variety is good, variety one, okay, this variety is good in compared to variety two. So I am telling the farmer to go for more on variety one. Okay, so they will go more profit. But sometimes the statistical error is there, so that null hypothesis will be wrong, or maybe the alternative hypothesis will be wrong, but the decision will be wrong. Okay, so that can be a make a totally different scenarios in terms of your decision. So that is called a statistical error. We will talk about it later. Now comes to the populations and samples. So what is population, what is sample, everybody knows, but a very simple way I can tell you that whenever you're going to sampling any things, we are telling basically sampling, you are not sampling all the individual in the population. No, we are not going, we are not collecting measuring all the fish in the pond. No, we are not measuring all the wheat or the rice in the agriculture field. No, we are measuring some of the part of the populations. Okay, so that is called some of the part of the population, that is called sample. Okay, what is sample? Sample is basically subset of the populations. Okay, we if the population is the whole big area, so we are measuring some of the part or randomly. What is the method? So we will see randomly we are collecting some of the sample from the whole populations. So this is the sample and populations. Now, as I told you that data is everywhere, it could be aviation sector, education, manufacturing, epidemiology, government, environmental, every sector. So decision is the most important from the data analysis. That is also we have already discussed collecting, organizing, presenting, analyzing, and valid decision. Okay, so this is the definition of statistics. Now comes to the types, classifications. So there is a two type of statistics. One is the descriptive statistics and another one is the inferential statistics. Descriptive statistic, by word you can say that this is basically describe. Okay, so it's basically describe your how your data is. Okay, so it's basically the summarizing or organizing or presenting data in an informative way. So we are basically describe how is the data is. Huh? we'll see later on also all of the things, maybe the mean, median, mode. Okay, so these are the basically descriptive statistics. Okay, or standard error, how the data look like. Okay, so this is basically how the even the distributions. Okay, so everything is comes into the descriptive statistics. Inferential statistics, it basically you have to do a mean or compare with the means basically. So we basically this method basically used in a populations and in sample, as I told you that you have to collect some of the samples, not the whole populations. Okay, so portion of the population is basically called a sample. Then we are comparing the mean with the populations. As I told you that our mean population's blood pressure is 120 by 80. Okay, but if my sample I found the blood pressures of this whole population is 140 by 90, then I can tell that these samples, okay, from these populations is basically this area is highly blood pressures having this one. Okay, so this is called an inferential statistic that will compare the mean with that your value with your data or maybe the populations. Okay, now, this is the descriptive statistics. Univariate, uni means 1, bivariate, bi means 2, and multivariate means plus 2. Okay, so multivariate. So univariate, shape of the data. How is the shape? Is it this shape? Okay, is it this shape? So what is the shape of the data? Then central tendency, we know the mean, median, mode. So the central is this here in the mean, or central is here in the median. Okay, so central of the data, central tendency, shape of the data, spread of the data. I mean, how is the distributed? The data is distributed from here to here. So this is called spread of the data. So mean to max. We know those things. Center means that standard deviation, standard error. Shape means kurtosis, skewness. I know you know about kurtosis and skewness. Okay, center, mean, median, mode. Okay, shape of the data is basically it's whether it's the mean and max. Okay, and the relative positions where it is. Okay, so it is here or here or here. Then bivariate analysis, we are basically compare two variable whether it is increasing with the other variable. Okay, so this is called correlations. Correlation is basically common example. I am telling that two variables you have. So one is increasing by the effect of others. Okay, so that is called what is the relation between these two, either positive or negative correlation. So ice cream sale increase if the temperature is increased. Okay, if the summer temperature increase, ice cream sale is increase. So that is called correlations. And what is called regressions? Regression is basically predictions. It's the form of correlation, but it tells you the predictions. So I am telling in correlations, the ice cream sale increase or decrease depend on temperatures. Here I can predict. If I tell you that today's temperature is 32 degrees centigrade, I can predict from this regression model the ice cream sale of this stall or this company will be might be 150. Okay, so I can predict with one variable to the other variable. I can predict the ice cream sale from the temperature. Okay, so that is called regression. And then multivariate. So if you have more than one variable, so that ice cream cell does not depend on the temperature only. It is also depend upon the humidity also. It is also depend upon the sometime any other variable also. Okay, company brand, whatever it is. Okay, so that is called multiple regressions. Okay, or fish catch. Fish catch is does not depend upon only the effort. Fish catch is also depend upon the water quality, maybe chlorophyll concentrations, maybe the pH, maybe other variables. Okay, so that is called multiple regression. It is not depend only a single variables. It's depend on multiple parameters. So that is called multiple regressions. And inferential statistic, as I told you, it is basically compared to means, as it can be compared to the means of the populations or means of the other variables. So here is two groups. If it is, then it a t-test. If you have a two variable, variable 1 and variable 2, then you have to do a t-test. Okay, if you have a more than two variable, variable 2, 1 and 3, then you have to do a ANOVA. So ANOVA is always basically 2 plus, and t-test is basically two variable. Okay, and there is a many more advanced statistics is there. We are not going to into the deep in this course. Now, data collection technique, it's very simple, everybody knows, but I'm just telling that it might be useful. So using available informations, whatever the available informations from secondary data. So it could be a earlier published any checklist or any governmental data depository or repository or any other form you can available information is still there from government reports. Okay, so that is called available informations. And second one is observing. I am going to field and observing how much is birds is there, what is the number of birds or number of fish, or measuring what is the length of the fish, weight of the fish. So measuring, so that can be one of the technique to collection of the data. Third is the interview. So I am going to field in with the fisherman, then I am interviewing with them that whether catch is increasing in last 10 years or not. So interviewing is also tell you is very essential in different types of science. It is not only social science. People are thinking that interviewing is also used in science, in social science, but is can be used in different science. So it is basically a qualitative science, whether it is good or bad, whether it is there is a lot of way to interview, and then questionnaire formations, and then different structures is there. And another other format of the administrative written questionnaire, that is almost the same of like qualitative data analysis. Now comes to the sampling methods. Everybody knows about it, but just I'm simply explain this one. Simple random sampling. It does means anywhere. So this is my area of sampling. I am sampling anywhere from this area. So that is called simple random sampling. I don't have any specific date or time. I don't have any specific area to sampling. It is randomly I am collecting the samples from the field. That is called simple random sampling. Now comes to the systematic sampling. It is by the name. So it is systematic. Systematic sampling means there is a system. So either I am collecting every first week of the month, okay, or I am collecting every that particular location. So systematically I am collecting the samples. Okay, so first week of the month, or maybe after 15 days, every 15 days I am collecting the sample. I am collecting 100 sampling. So there is a systematic way I am collecting the sample. Then stratified sampling. Stratified sampling is also divided into different parts: stratified random sampling, stratified sample sampling, different are. So stratified sampling is basically I am collecting sample from different strata or different populations. Okay, so here is one of the example I'm giving it you. So within the population there is a different groups. Okay, within a population might be different groups are there. So I am collecting the data from each and every groups. Okay, so then all the data I will analyze from this whole group. Okay, so that is called a stratified sampling. So I'm collecting data from maybe group 1, group 2, group 3, group 4, or maybe strata 1, strata 2, strata 3, strata 4. So different area I'm collecting. So it is not a one sample from a one group. Then multi-stage sampling, it is very simple. Stage multi, so multi means multiple, and the stage means there is a level. So here I am going to collecting sample from a particular area. Then I am collecting sample from a village level. Then I am collecting sample from a district level. I am collecting sample from a state level. Then I am collecting from a country level. Then I am collecting sample from international level. Okay, so I'm collecting sample in different stage. Okay, so that is called multi-stage sampling. Or I am collecting sample from different stage of the economic stage of the populations in a country, a low income group, a mid income group, or high income group. Okay, so I'm collecting sample from these three stage. So that is called multi-stage sampling. Okay. Now graphs, it's very simple. Everybody knows. No need to explain more. I'm not going to explain these graphs, but just I'm telling you some of the visualizations things. So graph is very essential in the data analysis because it will helps you to visualize your data. If I tell you the length of this of a particular piece or anything, I tell you some of the values 10, 15, then 20, then 30. This cannot make you any sense. Okay, so if this data, if you have presenting, so people will not understand that whether the or the productions, whatever it is, it's increase, decrease, anything. No, we don't, we easily we can't understand. But if you plot this data into graphically, you will find out that yes, yes, somehow it is increasing. Okay, so from graphical presentation is very essential to visualize your data. So to understand the trend, you can use a very good graphical representations, but you have to do a statistical test after it. Okay, so graphical representation is good, very good for the visual representation, but at the same time you have to do a statistical test to say yes, it is increasing or decreasing. Okay, so there is a different types of graphs in there, bar graph, pie chart, line chart, scatter plot, histogram, infograph, and nowadays different softwares are coming, different tools are coming. You can find out excellent graphs from any tool, free tools. Okay, so don't bother about the graphs, but important thing is that which graph you are going to use for which kind of data. Okay, I am going to bar graph for a percentage data? No. I am going to use a time series data into a pie chart? No. So that you have to know that which kind of bar graphs or which kind of data you are using for particular graphing. Okay. So here just if it is a vertical data series or time series, okay, time series, it's so in vertical data series you can use a bar graph or a horizontal bar or vertical graph, both in any country wise productions, whatever it is, it's very good representations of a horizontal graph in a in this way. So easily you can find out in a ranking first to last, whatever it is. Okay, so that is called horizontal and vertical bar graph. Then a segment means if my data is divided into percentage, so 100%, so it can be divided from three different sector or four different sector, from which sector it's coming. So very good segmentations you can present in this scat graph or any questionnaire or any 1 to 5 scale or percentage of the response. So that can be also very good representations in this graph. Here I told you that where you are not going to use a kind of graph. So in a bar graph, you cannot use when your data is very low, when data is very few data. It's not make any sense of this kind of graph. Okay. Pie chart is basically used for percentage. Usually we are using for percentage data. Okay. So in a population, how much male, female. Okay. So in a class, how much male, female, or good, bad, whatever it is. Any when there it is percentage comes, then you using basically pie chart. Line graph is basically used to any time series or whatever it is. It can be very good representations of increasing trend or decreasing trend. You can use a specific value and relationship between the variables. Okay, so that can be compare the trends of the groups, that can be used as a line graph also. Scatter plot is basically we are using every time whenever we have two variable x and y, and then we are plot firstly the scatter plot. Okay, so scatter plot is basically a way to identify that the trend and the another variable is dependent with other variable. Okay, so the basically the scatter plot is used for the regression analysis and also the correlation analysis easily. So this is called a regression line. We are plotting. It could be a nonlinear regression or linear regression. We'll discuss in a regression lecture. Then histogram, it is also very much useful for to identify the data type or data distributions. Okay, so it's basically interval scale, or you can use of uh, 1 minute. There is a lot of, I don't think so there is a. Okay, so this histogram basically used to tells you the shape of the data. It's very essential that the data is basically normally distributed or it is not a non-normal distribution. So how we know? Basically, the normal distribution data is basically like this. Okay, a non-normal distribution data is basically look like this or maybe look like this. So that can be very easily estimated from this histogram. And why this is used? Because of I have two type of test in statistical analysis. One is called parametric and one is called non-parametric test. We will discuss in later on also. So non-parametric, where you are going to do using a parametric test when your data is normally distributed. Okay, and when you are going to do use a non-parametric test when your data is non-normal distributions. Okay, it is not a linear or non-normal distribution basically. Okay, so here the median is the mean, and here is the mean is the mean in the population. So that is used for histogram. So this is about the first lecture, and then we are going to make the second one, the some statistical methods. Just a second. Okay, at this stage, do you have any questions from these first lectures? Yes, if you have questions, you can ask,
So
or
can
maybe
we,
yes,
sir, thank you so much for the first session. So my question is, you mentioned about the free tools for graphs. So can you share the names of those?
yes. I'll share everything. No problem. There is a
Okay,
lot of tools. I will send everything in the last email. No issues. Okay.
okay. Thank you.
As we have very limited time, 2 hours, we have to finish lot of lecture and also analysis. Okay, so you can ask questions, no issues, but yes, very short questions. So go to the next topic, that is the methods. I already discussed all the methods, basically very short, but I am going to discuss in little bit deep here. So statistical methods, as I will already discuss the descriptive inferential statistic. I hope you understood this thing: univariate, bivariate, and multivariate. Whether you are comparing means, you have to go for inferential statistics. Whether you comparing both the variables or bivariate or multivariate analysis. Now, this is very simple statistics. I'm going to tell you that everybody knows, but the most important thing is that we don't know about some of the times mean, median, mode. Okay, so mean, we know that sum of all data, and then you have to divide it with your sum. Okay, number of samples. So this is called mean. Mean basically very simple. Mean can be used, that is the important part of this. Here I told to you, the mean can be used when your distributions or your data is basically a linear. Your data is basically a not linear, your data is basically distributed on normal distributions. Okay, at that time we can, what is happening? You are not,
Another is maybe sharing his slide.
why, who is allowing?
But no, his skin is like not presentation mode.
So who is allowing to share him?
Oh, so I was trying. I didn't enjoy. Igbo nọ na nyanyi oge mgbọrọ bụ Igbo nọ.
Okay, now it's okay.
Yes, no, yes,
Okay.
absolutely.
So mean is basically we can use for when the data is basically normally distributed, and when we are using median, when that median will be used when the data is basically not in normal distributions. At
But we are
that
not
time,
seeing your slide. again.
We can see your slide, please. We can see your slide, please.
Yeah, it's not showing.
We can see the
Yeah,
slide,
it's
sir.
showing.
Okay, but it's not in full screen.
Baba, what am I doing you? Where are you going to?
It is showing, doctor.
Okay, okay.
I think the problem is the internet from there.
Hmm.
Dr. Sachendra, I cannot see the slides. I'm from Bangladesh.
Okay, just a
A
second. Just
slide is
let me.
not showing.
Okay, just a second. Just a second. Yes, now it's okay.
Yes, sir.
Yes,
Yes,
now
sir. Right. 1685.
Yes, now it's fine.
Okay, so mean is the basically the sum of all data. Then you have to divide it with the number of samples. Okay, and then next you have to go for median. I told you median is basically it is a series of data. Whenever you receive any sequence or series of the data, you median basically simple calculation that you have to arrange in a order from high to low. Okay, so ascending or descending order, basically ascending order you have to do arrangement, and then you have to find out which is the in the mean middle part of the number. Okay, so that way you can find out the median. But for a even number and or odd number, there is a way. Even number, it basically having the true value, and then you have to sum up or make average of these two value. Okay, and that way sorry, it can be arranged. First you have to arrange in a proper way, 2 3, then 5 6 7 9. Then you have to take the middle two part and then make a sum of this. So that is median of a odd number and even number, sorry, even. And for odd you will find out one. But in case of mode, you will find out multiple modes can be, or can be mode cannot be present in a series of number. What is mode? Mode is nothing but which digit or which numbers is maximum available in your series. Okay, so suppose in this series I have 8 three times. Okay, so the mode is 8. Okay, so but there can be a multiple mode. Okay, what is called multiple mode? This is unified or single mode, but it can be, if I have in this series, if I have 310 also, so the mode of this whole series will be 8 or 10 both. Okay, so multiple mode could be available in a number series, but median could be 1 and mean also can be 1. Now variability, as I already told you, range, mean and max. So what is the range of your data? Then variance, we'll discuss in later on. Then standard deviation, as we all know that how your data is deviated from your mean, how your data is deviated from the mean. Okay, so that is called standard deviations. And quartile range, it is also very important. When you will go through a box plot, this is basically a box plot, where you will find out a quartile range. So quartile range is basically first quartile, second quartile, and third quartile. So what are these? First quartile is 25%, second quartile is 50%, and third quartile is 75%. Okay, and you have 0 and 100, that is basically the mean range, minimum range of your data. And 100, basically it is basically the maximum range of your data. And then you have a first quartile 25%, second quartile 50%, third quartile 75%. What does it means? We will discuss in a box plot. Okay, so this is the quartile range. And then coefficient of variations, so how your variations will be varied from the mean. Okay, so range, as I told you, that mean and max. Then variance, it's basically simple sum of the mean. You have a observed series, and from the mean, so this is suppose the mean, and you have a series of variations. So from the mean, how it is varies. Okay, then sum of this one and total number minus 1. We knows that basically degrees of freedom. Okay, degrees of freedom df, degrees of freedom, that is we will discuss later on also. This is basically number of sample n minus 1 in general, but there is a different form of degrees of freedom is there, we will discuss in t-test or ANOVA. Okay, so here is basically variance means from the observations you have mean, and from the means each and every observations how much it varies. Okay, so that is called variations. And if you go for a standard deviations, it is almost the same. You have to do a square root of the variance. Okay, so you have to calculate the mean first, then you have to see each and every data point how it is varies from the mean. Okay, then you have to calculate the standard deviation from this variance. Okay, standard errors, it is basically a estimations of accuracy of your data. So if the standard error is very low, means your data is very accurate. What does it mean your data is not very large distributions, there is a not varies too much from the mean, not varies too much from the mean. Some of the time we find outliner. So if this is the mean, you will find out that some of the data is available here, outliner, that are called outliner. If there is a too many outliner from your data, mean your data is not distributed normally, so your data is basically uh, varies from a wide range. At that time you will find out a very high standard error, mean there is a some of the problem with this outliner, maybe sampling error, or maybe huge variations in the populations, something wrong. Okay, so that whenever you find a very outliner, so standard error very high, and that your data is not in good quality. Okay, so this is very very essential thing, standard deviation and standard error. Okay, then you have to be, maybe you can delete that outliner to make your data concrete, or maybe good in a shape. Now, these are the simple equations. I already told you the variations, standard deviations, and standard error. No need to discuss. Skewness and kurtosis. These are the two important things. Whenever you do a descriptive statistics, you will find out the skewness and kurtosis value. But what does these two means? Skewness is basically tells you about the shape of the data. Shape means I already discussed to you. This is the whether it is a normally distributed or whether it is a non-normal distributions. So there is a positive skewness or negative skewness or zero skewness. Okay, so zero skewness means your data is not varies from that mean. Okay, so it is not one side is very high, one side is low, it is not. So that is called, I will say you here, and kurtosis I will tell you later. So skewness basically varies from -1 to +1. Okay, so if you have a very high zero skewness whenever data is normally distributed, and when some of the data type is very high in this side, very low in this side, that is positive skewness, and negative skewness from this kind of shape you have. So skewness basically tells you the shape of the data. Now kurtosis is basically, it will tells you that peak of the data. What is peak of the data? So this is one data, and some of the time you will find out this kind of peak. Okay, so peakness. Okay, some of the you have a time series, and then in this time series you find out one very high peak one time. So you find out a very high peak, so that is called kurtosis value you can find out. So kurtosis is also varies from 3, basically 0, so greater than 3, less than 3, or 0 means it's basically no high, and sometime you will find out very high. Okay, and sometime you find out very low. Okay, so these are the three formations of kurtosis. It will tells you that peakiness of your data, and skewness tells you basically shape of the data. So positive skewness, zero skewness, and negative skewness. And kurtosis is also three type: plurikurtos, mesokurtos, and leptokurtic, mesokurtic, and plurikurtic. So leptokurtic is basically high peak, mesokurtic is basically a normal, and plurikurtic is basically lower. Okay. Confidence intervals, everybody heard about it, but today I just discuss you what is confidence interval. Confidence interval is basically a very good statistical tools to analyze your data range. Okay, so confidence intervals, as every times we will see that that is basically probability values 95% confidence limit we are telling, 99% confidence limit we are telling probability value. Okay, so we don't know what is 95, 97. We know, but I'm explaining. And sometimes we're telling 97%. What does it mean? Okay. So that confidence intervals, it basically tells you, if I draw a graph for you, so this is a distributions of a data, and from these distributions, these are the range. Okay. So this is that the 95% range. If any statistical test is belong within this range, this is the mean. So if it belongs to within that 95% range, so that is basically statistically valid. And rest 2.5% on the left side and 2.5% on the right side, that is basically out of the range. We are basically, that 5% range is not used for this analysis. So in statistics, nothing is 100%. Okay, in the reality, nothing is 100%. There is, sometime you will find out the probability value is 0.000000. Okay, 13 time, 15 time, that means 90.9999 time is you are confident. Okay, but it should not be 100% in reality or in numerical values. So what does it mean? So it means, if you take, suppose you are measuring a fish from a population. So you find out a mean length of the fish is suppose 150 millimeter. Okay. Now you are find out, you are calculating a confidence limit, confidence interval from this fish. So I have a 150 mean, and then plus minus confidence interval is 20. Okay. So what does it mean? It means, Abdullah, please, can you please take care of this? So 150 means 150 plus 20.
नाश्ता नाश्ता बनाओ नाश्ता खाए बहन बस
Okay, so 150 20 means 170 or 130. Okay, so this is the population's limit. This is called confidence interval. So what does it mean? Means if you go and measure the piece of that populations, and I am 95% time confidence, that the piece length will be lies between 130 to 170 millimeter. Okay, so you got my point. So confidence interval means, if it is 95%, means if you measured any piece from that populations, and you bring and you measure the length, and you will find out that length of this piece will be lies from 130 to 170. But 5% time I am not confident. So that is called confidence interval. 5% time it can be beyond 130 or 170. Okay, it can be goes beyond. So that is, in statistic, basically we are telling that 95% time confidence intervals, that is basically satisfied that it's with lies, all the values lies within that range. If it is out of this range, then it is not statistically valid. Okay, 99%, yes, it can be. So if it is, I am sure the 99% time the range between this one, it's depend upon the how good your statistical analysis. If the statistical values are very, so that means that your mean from that populations will be very restricted within that range. Okay, so that is confidence interval. You go through the equations, is very simple, as I already discussed this one. So 95% time sample proportions are lies within that interval, and 2.5% are it is out of the tail. It could be a right tail or it could be left tail. We will talk about that right and left in later on. Okay, so is everywhere it is there. So common example, I'm giving you the probability sampling. Probability, you know about it. So probability is basically a traffic line. Okay, so there is a people are crossing from here and there. Okay, so if there is a vehicle number is if one people is crossing this road, so what is the chance of accident? it's less. If two people is crossing this road, it will little bit more than that, mean 50%. If there is 1,000 people are crossing the road, so what is the probability of accident? It will be increased very high. So what does it means? What that means, if you increase your sample number, if you increase your sample number, if you increase your sample number, your statistical test will going to be valid always. Okay, statistical test will be always significant. Okay, so that is called probability. So if you increase your number, so if as you see here, if you increase the number of people, that accident chance of accident will be increased. Okay, so at that time you do a test and you have to put a red light here crossing, or you can put a so that is called decision. Okay, in statistical test. After the test, now we have a two type of things. One is the differences between the means, and one is the relationship. We have already discussed differences between the means is the t-test and ANOVA. So in the t-test and ANOVA, it's basically from the mean it will find out it's high or low. Okay, ANOVA is also plus 2. If you have more than 3 variable, more than 2 variable, you have to go for ANOVA. If you have 2 variable, you have to go for a t-test. Correlation, regression, I already discussed. Relation between the two variables, that is called correlation, and regression basically tells you about the predictions. Okay, so ice cream sale and temperature, remember this one. Now t-test, we have a two type of t-test we will discuss. One is the paired t-test and one is the unpaired t-test. What is paired t-test? Unpaired t-test. Paired t-test means I have two variables. Okay, so variable 1 and variable 2. So both the variable, if they are independent to each other, so that is also called independent and dependent test. Okay, so independent t-test and dependent t-test. So both the values are totally independent. They are not related. They are not related. Okay, so at that time we have to do a unpaired t-test. Okay, so basically, sorry, this is independent. Sorry, this is independent and this is dependent. And another variable 1 and variable 2, both of them are belongs to from a same subject. Same subject. Okay, so might be plant 1, plant 2, plant 3. You are taking the production value, plant 4, plant 5. So productions of this year, this variety 1, variety 2, or whatever it is, year 1 or year 2, then you're comparing. So both the values are coming from a same subject, then they are paired. Okay, so both the variables are belongs to same subject, then they are paired. They are dependent to each other. And when they are does not belongs to any of the subject, they are totally independent. This is independent, this is also independent. Then you have to do a unpaired t-test, or basically independent t-test. Okay, so t-test is also two type, dependent and independent. Now there is another variety is there, that is one tail and two tail. So one tail and two tail is basically, I told you we have a two hand, one and two. So what is this means? One tail and two tail basically tells you I have a two variety of banana, suppose. Okay, variety 1 and variety 2. So I am comparing between these two variety, but I don't know which variety is good or bad. Okay, in this case I don't know which variety is good or bad. At that time I will use a two tail test. Okay, I don't know, but I have a native variety of banana. Okay, this is my own variety in my garden. This variety I know its production is very good, but now I am planning to harvest a foreign variety. Okay, but I don't know what is it is good or bad. At that time I am comparing with that my variety, my native variety, with that foreign variety. I am comparing with that my native variety with unknown foreign variety. That time I will use a one tail test. Okay, so you got my point, how to use one tail test and when to use two tail test. Okay, now ANOVA or F test statistics, it is basically, I told you, it is three or more than three. Whenever you have you have a more varieties, variety 1, variety 2, variety 3, then you have to go for a ANOVA. So those are the same thing. It will compare with the mean, compare with the mean of all those three. So there is a three things in a ANOVA or F statistics. F value there, it will compare mean within, or it compare mean within, or it compare mean between the variables. So what is between? So it will comparing mean between v1, v2, v3, or it will comparing mean within. Okay, so I have, suppose these are the data 1, 2, 3, 4, whatever it is. So it will check the mean from between this version v1, v2, or v3, or it will compare mean between this to this, this to this, this to this. So this is within and between, that you will find out in a statistical result. We will discuss in that. But here is important thing, in case of t-test, you will find out that which variety is good. Any of the test, I can find out whether this good or this good between these two variable. So either one or two, so I can easily identify which is good, which is bad, okay, from this t-test. But in case of ANOVA, I have a three variety, so I cannot identify that which one is good, v1 good or v2 good or v3 good. I cannot tell you. Okay, so for this one you have to do a post hoc test. Okay, so that is basically used after the ANOVA test. It is called post hoc test, that can be used after the t-test. Okay, so after the ANOVA, sorry, after the ANOVA, what it will do a t-test after the ANOVA. So after the ANOVA, it will do a t-test between v1, v2, it will do a t-test between v2 and v3, and it will do a t-test between v3 and v1. Then it can tell you that which variety is good, variety 1, variety 2, or variety 3. Easily you can tell. So that is called a post hoc test. Okay. T-test comparing means, you have one dependent, one independent, both can be, and number of samples should be less than 3. Yes, it depends. And ANOVA, you can have a more than 2 or 3. So 2 or 3 independent variable can be, and there is a more sample can be available in the table. Now association, as I told you, correlations coefficient, as we all know, correlation coefficient are, okay, are this range values from -1 to +1. So what does it mean +1? When it goes to positive or negative. So positive correlations, so ice cream cell increase with the time, ice cream cell increase with the temperature. So if the temperature increase, ice cream cell will be increase. Okay, so that is a positive, and negative if it is decrease with another variables. Okay, so that is called positive or negative. So that's why the association, positive correlation and negative correlation, varies between +1 to -1. Okay, and there is a different format. R 0.3 means it's low correlations, and in case there is no relationship, you will find out R is equal to 0. Okay, so between this variable there is no relationship. Okay, and if it is increased little bit, you will find out how strong it is. It will find out a very strong linear line. Okay, so that depend our value. So R 0.8 or this. Now comes to correlation is also different. uh, Varieties is there. Basically, in general, we are using Pearson correlations. It is basically used for normally distributed data, and we have a Sparkman correlations. It basically used for any ordinal data, not continuous data. What is continuous? What is ordinal data? As I told you, continuous data is basically a normally distributed data. It have a continuous, and ordinal data, it have a order. It have a order. Order means is suppose strata 1, strata 2, strata 3, whatever it is, or stage 1 cancer, stage 2 cancer, whatever it is. Okay, so it has a order 1, 2, 3, 4, any order at that time, or rank is basically a rank. At that time you can use a Sparkman correlations, or if your data is sometimes non-normal distributions, you can use a rank correlation, that is called Sparkman rank correlations. In usually we are using Pearson correlations whenever our data is normally distributed, and it is a continuous. Nominal variable, for in case of nominal variable, means you have a age or marital status or sex, whether male female, yes no. Okay, this kind of, if you have we will do all the practice, or married or non-married, married or, so this time we will using pi correlations, kappa correlation for multiple variable. For two variable we are using pi correlations. And multiple correlation, as I told you, that if more than one variable is dependent on the other variable, so then you have to do a multiple correlations. Okay, so it's maybe I have a multiple parameters. Okay, it's have relationship between this multiple parameter, then you can do a multiple correlation matrix easily developed. So I have a parameter, suppose water quality, you take salinity, temperature, maybe humidity, or maybe such depth. Okay, water quality, so different parameters, you will find out what are the relationship between this temperature to humidity, what is the relationship between the such depth with the water quality. Okay, so this kind of multiple parameter you can use multiple correlations. But important thing is that we are usually using a correlations, and then we are write down the R value. Okay, R is 0.8, positive or negative, whatever it is. And then we are telling there is a positive correlations between this variable and this variable. Basically statistically it is wrong. Okay, and sometimes we are telling that R is equal to 0.7, even R is equal to 0.4, and then we are telling that there is a no relationship between this, that statistically not valid relationship between these. Yes, that is wrong, because this correlations is does not statistically valid until unless you do a F test for multiple and t test for single two variable. You have to do a t test between these correlations. Here I have mentioned. Okay, so you have to do a significant test for the R of t test, then you can find out. So R is equal to 0. Sometime what happen, it's if it is low event but it's statistically significant variations. Okay, significant. Okay, so that's why you have to write down, do a t test after it. R is equal to 0.56, and you have to do a t test p or probability value if it is less than 0.5. So then it is a statistically, you can tell that these two variable are positively correlated, significant positive correlated. Okay, but without this test or t test or probability value, you cannot say that these two variable are positively correlated significantly. Okay, now the third one, then we'll go to the lab. Yes, maybe 10-15 minutes I'll finish it. It is very interesting for who are going for a research, and we are planning for research also. So this one is basically,
Dr. Sachendra, before going to the next label,
yes,
I just want to ask you that are you going to provide us the slides, PowerPoint slides?
I think I explained several times. I'll provide it all the materials. Okay.
Oh, thank you so much. Thank you so much.
So here it's very interesting lecture, this one, that is experimental design and hypothesis testing, because it's very interesting. Why? Because most of us, when we are planning for our higher study, maybe my master thesis or PhD thesis, we don't know what to do. We are visit our guide, then we're telling them yes, this is this thing, then we are choosing our guide, then we have totally don't have any idea about the research. So research is basically means your hypothesis. Research is means your experimental design. Okay, research does not comes that yes, I have to do a PhD, that's why I have to do the research. No, research means you have a some questions should be come on your mind, that is called, we are talking about research questions. Okay, so research questions, if it is with your in your mind, then with that research questions, that you visit a guide that will be related to that field, then you hypothesize some of these research questions of that area. You hypothesize, I will tell you what is hypothesis. Hypothesize that yes, it is can be high or can be low, whatever it is, then you can do a conduct a research, and then you will do a test, then you will reach a conclusions. Okay, so that is basically a experimental design and research hypothesis. So experimental design, it's basically a form of study. Okay, whenever you have to find out a dependent and independent variable, very very crucial. Most of us, we don't know that which variable is dependent on others. So independent and dependent variable find out is very essential. Okay, then you have to find out a experimental design. It means that first of all some questions comes to in your mind. Okay, I told you. So some questions, some things that yes, one questions might be, I see in that area might be lot of pollution is increasing. Okay, so I thought I search, so then literature survey, these things will come. Okay, so you have to do some pre-experimental design research questions. Okay, so research questions should comes in your mind first, then based on these research questions you design your experiment. Okay, what I am going to do to measure that air quality of that region, it is deteriorated. So then I do some of the study literature review, that I find out that yes, it is some they're telling it's increase or whatever it is. Then you design your research question, how you do the analysis, everything. Then you have to find out a hypothesis. What does it mean hypothesis? So I'll tell you the steps. So formulate first research questions, then you have to do comparison. I told you it with the control, with the global control, might be the air quality, P 2.5 or P 4.5, whatever it is, particle matter. You can control with that global control. Then you compare with your result. Then to replicate, replicate. You have to do one to minimize the error, to minimize the error. Okay. Then randomization is very important, because what happens sometimes if you don't do randomization in your research, might be you will be biased. Okay, some of the common example I'm telling, I am doing a water quality of a water pond ecosystem, okay, or a river ecosystem, and I am going to sampling next to the debris, or next to the what is called going to sampling every time next to the effluent. Okay, a industrial effluent is coming and I'm going to sampling on that area particularly. So that is, you are not randomizing. So you are showing that yes, if you're measuring the water quality of a effluent area, I mean where the industrial effluent is coming, always it's a BOD or whatever, water quality will be very low. So that is called a biased. So you don't must have not be biased. You have to collect the water quality from everywhere, not next to this effluent. If you're going correct to this particular area always, so you are basically showing a bias. Sometimes it's doing lot of researchers, they're doing that, showing that yes, getting more fund. I am telling you that yes, water quality of this river is deteriorated. That actually this is not, because you are sampling next to this area, so it is always low. You are not sampling everywhere. So randomization is very important for your research. And then stratifications, I already told you, different block or different strata you have to collect, not only one area. And factorial experiment, you have to do different experiment, not in one factor. So the water quality does not deteriorate it for one factor. There is a lot of factors is there. So multiple variation is very important for the research. So you have to be unbiased, you have to be high precision as much as you to minimize the error. You have to be very simple, don't make any complex thing that cannot be possible to measure. You have to always, your research must have a wide applicability. Now if you send a research paper to a journal, most of the time they are rejecting because it's not a wide range. It is narrower, it is local, it is this they are telling and they are rejecting. Okay, so must have a wide range, and it is always uncertainties. So each and every time there is a lot of uncertainties. I told you, net environmental factors, then lot of political factors, then the different factors are there. So that replications, randomization is very essential to minimize the uncertainties in your research. Hypothesis testings is very interesting. You have to go through a step by step. I have give you one questions here. Okay, so this is because of my previous research area on that area, so that's why. So first of all, you have to find out the questions. What is my research questions? Here my research question is, I know how many of you know this fish or not. Hilsa is a very popular fish in this Bay of Bengal region. It is basically migratory fish. It comes to in a river when the salinity will be declined and come for the breeding. So here is my research question is that, is there any relationship between the Hilsa catch and seawater salinity? Okay, so this is my research questions. So now what I have to do, I have to formulate a hypothesis. Hypothesis means I am having, yes, might be there is a relationship or no, either or, there is a relationship or no. So I have to do a hypothesis testing, that is called two hypotheses. One is called null hypothesis or alternative hypothesis. In statistically, basically we have to prove the null hypothesis. Null hypothesis basically is always, it's tell you that there is a no relationship between these two variables. Okay, so it's tell you that there is no relationship between Hilsa catch and seawater catch, water salinity. Okay, alternative hypothesis, there is a relationship between Hilsa catch and seawater salinity. Okay, now you have to, what you have to do, you have to go to field, you have to collect the water quality, water salinity measurement for 5 6 years, whatever it is, or you can do a p historical data, or take the fisheries catch data from ministry or whatever it is, statistical yearbook, whatever it is, you take the catch data and you take the salinity data from satellite, whatever it is, wherever you collect your data. Now you do a test. So what kind of test I am doing here? I do a relationship. My question is, so that test is also depend upon your research questions. First of all, very important. Each and every time we don't know which kind of test we have to do. Okay, so that's it, totally depend upon your research questions. If you're comparing between these two variety, then it will be t-test. If you're comparing between three variety, then it will be ANOVA. If you find out the relationship, then it will be a correlation. If you want to predict something, I want to predict the Hilsa catch with the seawater salinity. Whatever the Hilsa catch will be next year, if the salinity will be this much. Okay, so then we have to do a regression. So we have to do a correlation test between Hilsa catch and salinity. Then after the t-test, I do a test, I do a Pearson correlation, whatever it is. Then I find out the relationship is very good, 0.78 degrees of freedom number of samples, you know, degrees of freedom, and probability value is this one. Then from this one you can predict, yes, so this is, I got my result here. Okay, so this is my result after doing the analysis. So now you have to do a decision. So what is my decision? My decision is I have to reject the null hypothesis. Why? Because my statistical test is valid. So it is within that 95% limit. It is not 5% limit. Okay, so you see probability value is less than 0.05, means it is within that 95% limit. Okay, I have told you. Okay, so you have to interpret, what is your interpretation? Yes, there is a strong positive correlations, mean if salinity salinity will seawater salinity, sorry, here it will be negative relationship, sorry. If the seawater salinity will be decline, then Hilsa catch will be increase. Okay, so there is a negative correlation between these two variable. If the Hilsa catch is increased due to decrease in seawater salinity. Okay, so this is the way, if you test your hypothesis or if you design your research questions, you will get a very good path in your research. Then I already talked about a null hypothesis, alternative hypothesis, just go through it. Hypothesis testing rules, I already told you. If probability value is greater than 0.05, you have to reject the sorry, less than 0.05 means you have to reject the null. Okay, you have to take the alternative, means there is a association, there is a association. And if the probability value is greater than 0.05, means you have to reject, you cannot reject the null, you have to say that there is no association between these two variables. Choosing the right test, it is also important. Which kind of test I am going for, very important. So it's depend upon your research question, first of all. Okay, it depend upon your research question, what questions you have formulated. Based on it, you have to choose the right test. So as I already discussed, which kind of test, when you are going for t-test, when you go for ANOVA, when you go for correlation, when you go for regressions. Okay. So as you know now, so research questions, it should be very clear and it should be measurable. Sometime I have a research question to measure the, suppose carbon dioxide concentration in moon. That is not possible. That is not measurable. So this unwanted or ambiguous research questions should not be formulated. So which is measurable, which is possible, which is clean, simple, always be simple. And then yes, just read it. Now choose comparing the mean. If you go compare between the mean between these two variable, you have to go for independent test if basically they are not dependent, and if between the groups they are not dependent, they are not coming from the same subject. If two, then t-test. If three or plus, then go for ANOVA. Comparing mean within the same subject, again and again I'm telling same subject. So both of the things are coming from the same subject, both the things. Then you have to do a paired t-test or dependent t-test, or in case of ANOVA you have to do repeated measured ANOVA. Okay. In case of associations, you have correlations and multiple correlation. If you have more than three variable, if you have two variable correlations, and for dependent and independent variables. So as I told you that if ice cream cell is dependent with the temperature or not, so temperature prediction, ice cream cell temperature can be predicted with the ice cream cell. At that time you have to do a regressions. More than three variable, then multiple regression. Suppose the ice cream cell is not only temperature, it is also humidity and other factors. Okay. So now the regression is also the linear and non-linear. I told you what is linear. Whenever you will find out this kind of relationship, this is linear. Some other time we will find out this kind of relationship, or can be this kind of relationship, or different format, different are there. We'll talk about it. That then you have to do a non-linear regressionship. It could be log format. Okay, it could be a square root format. Okay, it could be a what is called square root format, log format, different format are there for non-linear regressions. And correlation, Pearson correlation, basically whenever both the variable continuous, both the variable nominal, as I told you, yes, no, married, non-married, then two variable kappa, three variable phi, and three variable kappa. And rank correlations, whenever you are measuring ordinal data, order 1 2 3 4, or rank data, then you have to go for a Sparkman rank correlations. Variable, scale and category, you know about it. Scale continuous and discrete, and category is ordinal and nominal. Ordinal, order, there is order, and nominal, age, marital status, sex, this kind of things. Okay. So today we are almost finished these three lectures. Now we are introduce the lab session in R. I will show you the things. I hope most of you are not used to in R, but I will tell you how to download and these things. Very simple, very simple. And why R? Also, I'll tell you just in a short lectures. Just 5 minutes I'll take into this lecture, then I'll go to the practical session. So I hope everybody is nowadays learn about R, or at least know about R, because it's nowadays very popular in different sector, not only in data analysis, different sector. Okay, so R is a tools. It's basically used for different analysis, and most important thing is it is free. It is free. So whenever one thing is, I'm going to tell you that why I'm mentioning about it's free, and more importantly, because we are using Statistica, SPSS, then Stata, different softwares are available in the market. We are using pirated version, or different version is using. But basically this is in research ethics, this is basically illegal. You cannot use anything that is basically, you don't have the license. If you have the license, then it is okay, you can use it. You can write a paper using a pirated version of SPSS, that is basically ethically not acceptable. Okay, it is basically unethical. So if you have a license, then you can use this SPSS, stata, whatever it is, minitab, whatever it is. Lot of statistical tools are there. But if you don't have the proper license, you cannot use pirated for a research topic. Okay, so that's why R is free, and it both it can use as a programming language if you are very good in language, that is very good, and also it can be used statistical tool. But not only statistical tool, now R is used for every sector. Whatever is your topic, it can provide it you analysis. It's starting from the it can be species diversity analysis, it can be a medical science analysis, it can be a engineering science analysis, anything it can do. It's very good graphical tools. It is not a company, it's maintained by a lot of people, it's a group of people. They are basically, it can use any platform, Windows, Mac, or any platform it can be used. And it's a continuous process, it's going on. So where you will find out the R, you can just log in rproject.com, then download the latest version, install on your system. It does not require a very high configurations, it can run in a simple computer also. So from rproject.com, you can .org, you can download it and install on your system. But R is a the background software. Now due to the user friendly version of the R is coming to R Studio, that is much more user friendly. So but to to run R Studio, you must have to install R in your system. So first you have to install R in your system, then you go and download R Studio from the system, and then install in your system, then work in the R Studio. R Studio is basically very much user friendly. It can comes but too much user friendly. Okay, so we'll see later on when you will do the exercise. Okay, so first you have to install R, then you can install R Studio in your system. So both the link are available here. You can go in Google, just type down R, you can install it, and then type down R Studio, install it in your system. So it is the interface of the R. There is a all the comments is available, and it's easy, very easy, and it comes to automatically. So and it's very good, and the management of this whole software is very user friendly, as I told you. We'll see what is there. There is a four part in a R Studio. There is a code editor where you can edit your code. Okay, then console where you get your result and all the analysis, and environment or working sheet. Here you can see your history, you can see whatever the variables is uploaded in the system, and then you can upload or your sheet from here. Different things are there. So that is working environment or history. All the variable history will be here, and here you can find out your all your graphs or your plots or your graphics will come out here. And you can find out here a little tag that is called package. That R Studio is required package. Package means each and every analysis has combined with a particular package. Package means all the codes for that particular program, it's available on that package. So you don't have to go to the whole codes. It's combined in a particular package, compact, and then it will give you with simple code. Suppose species diversity analysis, so you just type down diversity, so it basically tells you that diversity package have all the predefined codes, all the analysis within that package, so no need to go. So particular package is very essential to install all each and every analysis part time. So first you have to go to a project. In I will show you the interface in the project. Then you can create a new project, and within the new project you just create a new directory in your system where you want to go to save your whole project or whole analysis in a single directory. So then in the file, then you will find out a new file, and you can find out a new script or script that you can type down all your code and save it. It is one time investment, then it will be used for any analysis for later on also. So R script you can generate your all the codes. Then different variables working function is there, ls different function are there. If you used to it, just go and start working on r. As I told you why it is important. Nowadays, uh, uh, each and every higher study organizations or each and every company, even job, even higher study job university, they are looking for any tools. Okay, so whether you are good in particular tools, they, that is hidden, that's not they will mention you. But if you mention in your cv that I know r, I know python, I know qgis. Okay, so those are the things. Any tool, learn yourself, start learning. Okay, any of the tool that will help you in your future careers. Okay, not only that, for higher study, also in job sector, everything, every company, even every higher study organization, they are also hiddenly they are looking for this kind of tools, you know or not. Even in the foreign university, if you apply for them, they will directly mention that our knowledge is required. Okay, or python knowledge is required. Okay, so that's is these are the tools is very essential to start learning from the beginning or maybe in advanced stage. But r is very case sensitive. Sometimes it's always type in the lower case, always in the lower case. So if you type in higher case, it will say give you a error message. So here I'm not going to explain these other things. It can be used calculator, different vector, you can use x. So but the simple thing is that you have to assign something. Suppose x I am assigning. Okay, so on that way I am assigning x is equal to 5. So if you type x, then automatically it will comes 5. So that is called assigning. So I am assigning a equations x is equal to suppose y plus 7. Now I am giving y is equal to 2. Now if I type x, x 2 plus 7, it will gives you 9. So that is assign how. These are the simple things. I'm not going to give in details. The length, mean, median. This is simple code, a summary of your result, difference, revisions. uh, then data frame, those are the because it's not a art training, but you can, there is a lot of source available in the google or what is called in youtube. Just start learning from the beginning. So data frame, you have to how to arrange your data in a frame. Matrix, it's different matrix, array, table, you have to, then the list of your data, then factors. Okay, so these are the very simple things in the R. Variables, vectors, okay, and then matrix. You can how you can go through a 4 by 4 matrix, 3 by 4 matrix, whatever it is. And array, uh, you can find out. So how to arrange your data in a frame. So these are the very simple things. I'm just going through graphics. Some of the point for the graphics: plot, points, line, text, legend, box, color, whatever it is. Just these are the code. For t-test, you have to type then only t.test. For variation test, you have to Fisher variation test, you have to var.test, correlation test, you have to, the code is core.test, chi-square, the chi-square dot test. These are the code in R. Okay. Now, some of the models, the linear model. So it's the Gaussians. So Gaussians means there is a two type of distribution: Gaussian distribution or non-linear distributions. So LM is basically called linear model. LM stand for linear model. So it's basically tells you that regression model. So it's or Y explained by X means Y. Y means the ice cream cell. It's explained by temperature. Okay, so this is x and y. Okay, and then multiple variable if this is y, the ice cream cell, it's depend upon the temperature and humidity. Okay, now if as a factor, you can make as a factor. Then linear models you can do for the ANOVA also, and also can be used for regressions. GLM, generalized linear model, is also one of the format whenever you have Poisson distributions or Gaussian distributions. Poisson distribution is basically nonlinear distributions, and Gaussian distribution basically the linear distributions. And output, how you get your output, lm dot out summary, plot, fit, whatever it is, you get the output of your result. GLM, I already told you, binomial or Poissons and Gaussian, these three distributions. You know binomial distribution. Binomial distribution, bi means two, nominal means yes or no. So yes and no. At that time, if you are very male or female, okay, at that time you have to do a binomial distributions. Poisson distributions, it basically gives you a non-linear or non-parametric test, Poisson distribution, and linear is basically Gaussian distributions. Okay, now we'll start in R. So before that, we have 25 minutes, but I try to finish it within time, or we can extend because it is one time. I will show you. It's easy. Code is available. You can try yourself. So 5 minutes I'm taking. If you have any queries or questions regarding the theory part, we'll discuss a little bit. Then we'll start the practical session. Any questions? Yes, yes.
Good evening, sir. So
Hmm.
previously you mentioned that for the correlation test, after getting our result R, we have to do the T test for its
Yes.
significance. So, sir, can you
Yes, it
briefly
is
explain
basically
that
the
how can we?
general, it's a general formations. So if you do the t-test in Excel, or not t-test, sorry, correlation in Excel, it will tells you whether the relationship is there only. It does not tell you the p-value, probability value, or the after the t-test whether it is significant or not. But if you do in any other software, even in R, just do the code dot test, it will tells you everything. You will get your result along with the probability value, means after the test also it will tell you. So whether your correlation is significant or not, you can get in a single test. No need to do other test. Okay, so it is within that test, chord dot test, within that test it is involved, that it will tell you that whether your correlation is significant or not. Okay, but in Excel basically it does not tells you. Yes, any other question?
Thank you so much, sir, for these clarifications.
Hmm. Any other question? Yeah, all the lecture I told you, everything will be available on Google Drive. Yeah, significant test between the child weight 50 and 60 kg. So it does not tell you between only two number. Okay, so you have a group of child weight. Okay, so you have a suppose 10, 15 children, their weight. Then you can measure the group in a two group, maybe class one or class two. Then you can compare the weight between these two class. So one child is 50 kg, another child is 60 kg. It the test will not tell you between these two only two number. Okay, so if it is done, then we will go to the next step. That is the practical. As I told you, I have told you to install R Studio, but first you have to install R, then you have to install R Studio. So whenever in the, I hope if some of you have the software, it's okay, and some who don't have still
Hello,
installed,
sir.
you.
Am I audible?
Yes, 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 yes.
Sir, you explained very well, sir. But I want one example in linear regression and non-linear regression, sir.
Yeah, I already I when we go to the regression, we'll talk about it. Linear regressions is basically uh, when the variables are linearly increase. Okay, so just suppose it's depend upon the equations of the relationship between the variable. Okay, suppose I can tell you that one in general population dynamics or a prey-predator interaction. Prey-predator interaction, what happened, that is not a linear relationship. Okay, prey increase, predator decrease, prey increase, predator decrease. So it's not a kind of a square root function or kind of a non-linear function. It is not a linear function. So if you want to do some regression analysis between the prey and predator, that time you have to do a non-linear relationship, non-linear regressions. Another example, piece length and weight. Piece length and weight does not increase in a linear way. Piece length is piece weight basically increase in a cube length way. Cube length. Okay, so if piece length is increased 3 millimeter, it cube of 3 means 3 into 3 into 3, 9 time it will increase its weight. Okay, so that's a cube length. So that is basically you will find out a power function. So that is a non-linear regression. So it depend upon the basically the which kind of analysis you are going to do. Prey-predator analysis, prey-predator relationship, it's not a linear. Piece length weight relationship, it's not a linear. So these are the example of non-linear regression. But ice cream cell and that temperature, it is a linear regression, I can say. And the other also temperature, humidity, that is also linear. If temperature increase, humidity also increase. Rainfall, humidity, these are the basically linear relationship. And some of the natural things that can impact with other factors, that sometimes it's not a linear regressions. Okay, it can be log format, it could be square root format. We'll discuss in the regression time. Okay, so here we you are showing, I told you that's a code editor. So in the upper left you will find out there is a code is written. Some of the code is already written here. Okay, so these all the codes I will share you. So first one is descriptive statistics, second one is the box plot will show, then t-test, ANOVA, all the codes are already written there with representative of the data, with respective data. So here this is the code editor. How you go to code editor, go to file and go to a new file and go to R script. If you type R script, a new R script will be typed, then you can type down all your code. Okay, so this is R script. Now how to prepare your project. So here on the right, I can, so you can go in right, you can say it's a project. It is none. I cannot save any project because it's a used. So here you can go and click a new project, and you can click a new project on your directory. You can use your desktop or whatever it is working directory, create a project and then save it. So everything, all the analysis of that particular project will be saved on that particular folder or particular area. Otherwise, by default it will saved on your C drive, or in C drive there is a R file is there. All the things will be saved on that one. Okay, by default. But if you want to make your own project, you can create your own project. Okay, so here you can see some of the codes are written here. I'll show you. You can drag all the things here and there as per your things. Availability. Now on the right side, I told you there is a package. If you click on the package, you will find out the list of the package, all the huge list of the package. So which packages are in used, there is basically marked, and which packages is not in used, there is a not in marked. So how to download the install the package, that I also told you. Okay, now if each I told you, each and every analysis is have a particular package, whatever it is, a bird analysis or a microbiological analysis or medical analysis, every package are available there, I think so. Okay, so there is a almost I think 100,000 packages are there available, not maybe 5,000 or 7,000 packages, but it's having all. So you can how to install, you can install, but you have to know the particular package for particular analysis. Suppose diversity index, I think diversity is the package. So if you type diverse or something, diversity or diversity forest or different package, you can see. Okay, diversification, so different package is available for this particular topic. uh, so you can find out the package automatically, then you can click install, it will be installed on your system, then you can use that particular package. So here for this particular descriptive statistic analysis, I need a package that is called installed dot package, that is the code to install read excel. But read excel, so read excel means it will read excel from my file. I have saved all my excel file in a folder that is saved there, my data. Basically you can import data in text format, you can import data in txt format, or import in csv format, or even any other format. I can show you here also. On the right side you can find out a set that is called import data set. So you can find out the format: text format, excel format, spss format, sas format, and stata format. So different format you can import your data set. But usually we are using our data in excel, or maybe in csv format. Okay, so that you can, for the read my excel, I have a package that is called read excel. So I have to install, it is already installed in my system. So each and every time you have to type your codes, then run one by one. So library of the excel, what is library of the read excel? Library means whatever available within that package, that will be unpacked, that will be unpacked for used in that platform. Okay, so that you have to see what is there in the library. So I have to do library in the package, means whatever available within that package, that is unpacked now, you can use that particular package now. So I have assigned data ds. This is the anything you can assign. That data ds is belongs to that particular data format, data file, that is available in my c drive. User, my name in the desktop, in statistical training, in statistics, then lab exercise excel, then the file name is descriptive statistic data dot xls. Okay, so how to find out the path of this way? So if I open, I think I have to, just a second, I have to make it 1 second. Stop presenting, then present entire window, screen 2, there. Yes. So how to find out the path of the data? So if I open if I open my whole system, if I go to my desktop, in my desktop there is a statistical training. Within the statistical training, I have a statistic file, then I have the lab exercise, then I have this data descriptive statistic data. This is the file. So if you click on the data, then on the upper this corner, this window, you will find out, if you click it, you will find out the path. Okay, so C user is desktop, this is the path. Then copy this path and paste on your code. Okay, paste here until the data file name, because data file name is already same, so you have to paste it here. So whenever you paste it there, you will find out all the slash are in the right or the forward. So you have to make it backward slash. Everything should be make backward slash, because R cannot read the forward slash, so it can be read sorry, backward slash, not forward slash. So you have to change all the slash into a forward slash. And experiment excel here. Now if you run it, yes, my data is came as a variable here. Okay, so if you want to view data, next is the view data. If I click on the view data, run it, you will find out this is my data. I have a length and weight of some of the fish. Okay, so this is my descriptive statistic data I'm using here. So this is my data. You can see your view data. Now you have to attach the data. Why attach the data? If you attach the data in the platform, so it can access everything whatever available within the data table, data Excel. So it is already attached. Now you have to structure of the data. So simple code I'm going to one by one. So structure of the data. It's if you run the structure of the data DS, you find out the data DS. I have assigned data DS that my data. Okay. So if you see the structure of the data, you will find out that there is a 50 data set into two variables. One is length and one is weight. 1 to 50. Both are numerical variables. Okay, not characters. It's numerical variables, and the values are here. Okay, so this is the structure of the data. Now if I click on the summary of the data, if you click, you will find out the basic summary. So minimum of the length is 160, first quartile. Now first quartile, second quartile, third quartile, median, mean, third quartile, and maximum. This is the things I'll tell you in the box plot. Okay, then weight, this is also minimum, first quartile, median, mean, third quartile, and maximum. So this is the summary of the data. Now if I tell you the mean length of the populations, I click mean dot length. The mean length of the population is 175.38 millimeter. Median, if I click, median is 47.44. Okay, then mode, if I click, there is no mode. Okay, so none of the data is repeated. There is no mode in this data series. What does it mean? There is no values in this number series, in this fish length does not have the similar data. Okay, now variations, if you click on the variance of the data is 23. Standard deviations is variance by square root of n. So, variance by standard deviations mean weight, standard deviations of this one, it's already mentioned. Now if you want to kurtosis skewness, there is one package you have to install, this E1070. Directly you can find out the kurtosis of this value and kurtosis and skewness. It says you negative skewed little bit, and kurtosis is not that much high. It is values is not 3, so it is bureaucratic. Minimum, maximum, length, range, different formats are there. Now there is a one package that is called Psyche. It is very good. Lot of analysis you can do using the Psyche, but that package tells you if you describe your whole data set, all the statistics in a single click. All the statistics. Now I am doing everything in a one by one. Mean one click, median one click, one code, kurtosis one click, skewness one click. So but if I know everything in a single click, now just install that particular package Psyche, library of it, and describe this Psyche, run it. You will find out everything in. So variations, then n, mean, standard deviation, median, then trimetric, median, mean, maximum, range, kurtosis, skewness, standard error. Everything in a single click, you will find out the descriptive statistics. Now histogram, I told you histogram is also one of the important thing that can tell you that data shape. So if you run the hist dot, then you can find out this data is almost linear distributed. Okay, you can see it is not a positive or negative skewed. It is almost a linear, you can see. Okay, now if you see the histogram of the weight, that is also highly linear. Okay, so it's a not linear, it's a means parametric test you have to do for this analysis. Okay, so it's a normal distributed data from the histogram you will find out. Okay, now box plot, I told you that box plot is important. First quartile, second quartile, I will explain here. So if you run the box plot, you will find out that. Okay, so in a box plot, what you find out here? So in a box plot, you find a dark line in the middle. So that is my basically mean. Okay, so that is my mean. And then you find out a box. So within the box, that is basically 25% quartile. Then the dark line, that is the 50% quartile, and upper line, that is called 75% quartile, and lower line is called, that is minimum value, and upper line, that is called maximum value. Okay, so if you go to this summary data, you will find out the minimum 162. If you go here, it is 162. Maximum 189. If you go, it's 189. First quartile was 171. If you line here, first quartile is 171. So that means first quartile is means 25%. So what does it mean? So if I take any sample from the populations, I will sure the 25% sample will be lies between 161 to 171 to first quartile. If I measure any space 50% time, I am sure that your values will lies between 161 to 170, what is called mean 175. Okay, 75%. This, if I measured any phase, 180 to 161 will be the 75% of the populations will be lies between these things. And if you measure from full quartile, mean 0 to mean to max, so all the phase will be lies between this one. Okay, so 25% phase will be lies between this phase, 50% phase will be lies between this length, 75% length phase will be this length, and 100% will be that the whole mean to max. So that is called quartile. Okay, now is this I am telling each and every time about Shapiro test. Yes, I'm going to tell you Shapiro test each and every time to tell you that normal distributed non-normal distribution. How to do the this test is, there's a lot of ways. So this is also one of the way, box plot. If you see that dark red line or the mean line, it is below par here, below par, mean it is not very far to the lower quartile. Okay, so that time it is a median, mean I can say from the box plot it is not normally distributed. Or if it is maybe above here, that is not normally distributed. And sometimes you will find out dot out of this range that are called outliner. Now Shapiro test is basically used to test the data normality. So what is normality? Is that normal distributed or not? So if I do the Shapiro dot test of length, if I run it, I will find out the test is give me a normal distributions. Why? The probability value, the Shapiro is, I already mentioned there in the text is there. So if the Shapiro null hypothesis is that your data is normally distributed, that is null hypothesis for the Shapiro test. So here my probability value is 0.6, mean it is more than 0.05, means I have to accept my null hypothesis. So what is my null hypothesis for Shapiro test? That data is normally distributed. So from one test, Shapiro is very effective. You can say that your data is normally distributed or non-normal distributed. So why each and every time I'm telling normal and non-normal? Because each and every statistical test will be varies. So I, instead of t-test, I have to do a Wilcoxon test. Instead of parametric and non-parametric, I'll discuss in the later on lectures next day, tomorrow, parametric and non-parametric. So that's why we have to check the data normality first. Okay. So data normality is very essential before run any test, because if you do a parametric test for a non-parametric data, that is statistically wrong. Or if you run a non-parametric data or a non-normal data in a parametric test, that is also wrong. So first of all, you have to check the data normality. Okay, so that's why it is very, very important, shape of the data. Then you can say, so here is a code to do multiple Shapiro, multiple variable Shapiro test in a one click. I have a 5, 10 or variables. I want to check each and every variable whether they are normally distributed or no. So you can using this code df this one. So you can find out both the data. You will find out that are normal. Both my length is also normally distributed and weight is also my normally distributed. How I can check? There is also another way to check the QQ line, the distributions. So you can say the QQ norm. Yes, and place the QQ line, you can find out that my length data is almost linear distributed or normal distributed. It follows the normalities. Okay. So this is the some of the things about descriptive statistics. First, you have to check your data structure. First, you have to check your data shape using box plot or histogram or Shapiro test or QQ normal or QQ line. So you can check the data is normally distributed or not. If it is non-normally distributed, then you have to do a non-parametric test. Next class we will discuss the non-parametric test. Or if your data is like this normal distributed, then you can do a parametric test. Okay, so this codes is very simple, but it's one like lecture. To our lecture it is not possible to learn everything, but as much as I possible I can give it to you. Okay, now I can show you one box plot. Multiple parameter box plot is very one of my student did it. Okay, so I'll show you one very interesting data of water quality analysis here. So here we need few more package in this water quality analysis box plot. I need a tidyverse, it's a very big package. Then I need a ggplot, very useful for the graphics. Okay, ggplot is very useful for the graphics. I need to install it and then I need to deploy. These are the very good package that can be used for very good statistical analysis. Now I'll see my data. If you want to see the data, I'll show you here. So this is the data of water quality of 4 different sample location. One is agriculture field, one is industrial field, one is urbanization area, and one is the landfill area. Okay, so different area we are collecting the water and then we are measuring the water quality. Then we will find out the water qualities of the different regions. Okay, so we have a measure different water quality here, that calcium, magnesium, TDS, pH, EC, different things are there. Now we will do a box plot to find out lot of things a box plot can tells you. Okay, so first we need to say this is also attach the data. I am attaching the data. Unpack one of the more so describe the data. Yes, you can see the all the describe kurtosis cueness of the whole data series. Now core plot correlation I'm not going to tell you now, so I'm not going to this one directly. I'm going to this yes here to these 3 4 lines. These are the used for multiple parameters, multiple factors in a single box plot. Very essential and very good, catchy for publications. This is very simple code you can use here. So you have to unpack a library that is reshape. Then you have to arrange your data based on different variables. I have a 13 variables of water quality based on sample locations. So it's very simple. You can see d of a, d dot a, I am arranging the data in a series that I have a 13 parameters. So data water 1 to 13, and ID means it will be divided into 4 sample locations. Okay, so now I run this one. Then I'll run this data in a frame. Then I run this simple code to make a multiple box plot in a single way. Okay, so if I run this one, you will find out a very good uh, graphics here. So let me explain this one. Is it visible? So I think
Yes,
it
sir.
is. Yes. So here you can see that all the sampling locations, agriculture, industrial, landfill, and urbanizations with all the water qualities. So here you see that EC. From here, one thing is it's not only one thing. There is a lot of statistical analysis can be told from a box plot. Box plot is very important, very important. So you see the EC. You see EC have 4 different landfill. EC is totally different. Okay, So mean EC for agriculture is around 4.5. Mean EC, electron conductivity, okay, electron conductivity of that industrial is almost 3.2 or something, or landfill is almost 5.6. Even here is also urbanization is also, but there is some of the what is called outliner in agriculture and urbanization, but it's okay, no problem. But from here I can say you directly, without doing any test, I can tell you that there is a significant variations in electron conductivity in four locations. Okay, I can tell you. So if from till now what we learned from here, if I told you that which test you have to do, which test you have to do to find out that statement, my statement is, my hypothesis is, there is a significant relationship, significant variations in EC, mean electron conductivity, in agriculture, industrial, landfill and urbanization area. Which test you will do? Tell me. Why t-test? T-test is
Sir,
for
can you
ANOVA
kindly
test.
ANOVA
repeat?
ANOVA test.
ANOVA. So
ANOVA
t-test is
test.
for two variable. T-test is only for two variable. Here are how many? I have four: agriculture, industrial, landfill, and urbanizations. So four area. What is my variable? My electron conductivity. If I check, so if I do run a, I have already done. I think so. Yes, I have in line here. You see that I have run a model one, ANOVA. AOP is the ANOVA data water of electron conductivity or in sample locations. Okay, so if I run it and if I will find out my result here in the down, you will find out that yes, it is highly significant. You see the probability value is almost telling e to the power minus 15. Okay, so it's 0.000 means almost 99. 3 star, there is also one statistic I tell you, that's one thing. So 1 means it's 1, 0 means 0. Here is a 3 star, mean it is 99.99% significant. Okay, so its probability value is less than 0.001. Probability value is 0 point less than 0.01. This one means it's 99% significant. Okay, probability value is 0.05. It means your probability value is 95% significant. Probability value is 0.1. What does it means? Means 90% significant. Okay, so we are basically considering above 95%. Okay, so here the sample location wise EC value is highly significant, that is almost 99.99%, or probability value is 0.001% significant. So from the that box plot you can say huge variations are there. Huh. So box plot is very essential for statistical analysis. Okay, so here is the very simple code you can run and you can find out a multiple parameter box plot in a single plot. Okay, now I'll show you t-test. I'll finish it in 10 minutes. Okay, so t-test is very simple. Here is the all the package, all the data is there. I'll show you that data one time. Yes, here I have three data in single field. Okay, so first one is the systolic blood pressure, that is the one sample t-test, one sample t-test. Then I have a field one productions. So these are totally basically unpaired field two and field one productions of that particular crop. It does not belongs to any of the particular plant. Okay, so it's totally unpaired. In third one, we'll do one paired t-test, that is basically first year and second year yield of different plant. So which year is much more productive, first year or second year? So here the first hypothesis is the systolic blood pressures. Population blood pressure I already told you 120. Here we have some of the data. So we have to check that whether this population is suffering from higher blood pressure or not. So we have to do a one sample t-test for this analysis. One sample means you have only one sample Okay, there is no other. You are comparing with that sample with the population. You are comparing with that one with population mean. So here one sample t-test code, you will find out this one. So t.test systolic blood pressures. Mu, what is mu? Mu is the population mean. Mu is the population mean. So here mu is the population mean is 120 systolic blood pressure, and I am telling the confidence interval at 95% level. Okay, means probability value 0.05. I can use it 99%, no issues. So then I can run this one. Just first attach the data and then I have run this one. So I got my t-test result, one sample t-test. Here you will find out t value is 1.24. Okay, degrees of freedom 14 means I have a 15 individual. So 15 minus 1, degrees of freedom is 14. And probability value is 0.23. 0.23. What does it means? Means it means that the I have to reject my null hypothesis, so accept my null hypothesis. So that population is basically not suffering from a high blood pressures. Okay, so its alternative hypothesis was that true mean is not equals to 120. Yes, I have to reject it. Alternative hypothesis, I have to accept my null hypothesis that the population is a blood pressure is equals to the population. I mean that sample blood pressure is equals to the populations. Okay, you can see the mean population blood pressure is the 123. It is almost same to the population blood pressure, that is 120. So from this result, I can say that this particular sample area or that particular population, whatever it is, they are not suffering from a high blood pressures. Okay. Now, if I go for a two test, first paired, I told you that paired data, if you've seen here, first year yield, second year yield of different plant. Okay. So if I run this t test, so what is the code? T dot test, two variable, first year yield and second year yield. Mu, I don't know, population mean. I told you that I don't know. Paired t test. So paired t test is true. T means true. Paired t-test is false if it will be unpaired test. Okay, and variance are equal, that is also false. Variance are not equal, so you can change the variance in the later one. Okay, line 26 you can check the variance of the two variable. Okay, so variance is not equal and paired is also true confidence level is 0.95. Then you run it, then you find out a paired t-test. So paired t-test, you will find out that probability value is 0.0002, means it is significant. So I can say that that between these two variable, between these two variety, there is a significant variations in the yield. Okay, so first year yield is significant different from the second year yield. Okay, so mean differences is 0 minus 67, but there is a significant differences. Okay, so that is a paired t-test. Then you can run a unpaired t-test on line I think line 17. You can run a unpaired t-test line 17. So run it, you will find out unpaired t-test. That will tell you yes, it's not significant. So what is my data? If I go to the data, so field 1 production and field 2 productions, so there is a no significant variations in the field 1 productions and field 2 productions. Okay, so that is my conclusion from this t-test. You can see the mean values is 18.45 and mean value of the field 2 is almost 19, so almost same. So there is a no significant variations in field 1 productions and field 2 productions. Okay. So this is the different type of t-test: one sample t-test, paired t-test, unpaired t-test. And if you want to do the one-side test, then you can mention that alternative is one-sided. Here is this one, this code alternative is one-sided, and you can tell you that what is the variation are equally varied or not. You can run from this var.test. You can run this code and then you will find out the variations are equal or not before running var.equal f or not. F or f means false and t means true. So you can run it and then you can plot it here. Okay, so this is the t-test. And then ANOVA is also simple. I'll just quickly I'll finish it in 5 minutes. So I'll click the data, just see the data in ANOVA. I have a three plant variety: plant A, plant B, plant C, and their productions. Production in kg. So I want to find out which plant variety is good: plant A, plant B, plant C. So I have to do a test, ANOVA test. So I'll do a data in a frame, then run the productions, mean productions in plant variety. So code is AOV, means ANOVA productions and plant variety. So if I run it, I will find out the result in summary. So here you will find out that productions between this plant variety is significant. So there is a significant variations in plant variety, plant A, B or C, or plant 1, 2, 3. But I told you from here you cannot say where is the significant variation: 1, 2, 2, 3 or 3, 1. Which variety is good? For this one you have to do a Tukey's HSD test, or you can plot the box plot to see the differences. I told you, yes, you see the mean differences from plant A, plant B, plant C is totally different. Plant A mean production is almost 220 or 210, plant B is almost 250 or 245, and plant C production kg around 270. So from here you can say the plant C is much more productive. But also you can do a Tukey's HSD test that will also tell you that between plant B and plant A, you see a line here, there is no significant variations. So plant A and B, no significant variations. But plant C and A, there is a significant. A and C is highly variations. So C is almost 230 to 70 and A is 210. So there is a significant variation between plant A, plant C. And plant C and plant B, yes, there is also significant variation, but that is not that much significant in compared to A and C. Okay, so this is ANOVA test, and you can do different other ANOVA also. I can show you 2A ANOVA. This is also important whenever you have a variety, means one independent and two dependent. Okay, so crop production is not dependent on the variety. Okay, I have three variety. I'm talking about now that variety A, variety B, variety C. Okay, so variety is not an important, and along with this, fertilizer is also important. Which fertilizer you are giving? Okay, so this is a two-way ANOVA. So I want to see that production is varied, belongs to different variety or not, or different fertilizer or not. Okay, and then you can find out the interaction between variety and fertilizer. So because corn production is not only depend on the variety, it is also depend on the fertilizer. So both the effect can be. So that can be one. A repeated measure ANOVA is also here. You can run, and I think today is almost we are going out of the time. So next day we'll finish this one, and then I'll give all the codes to you. You can run and you can use with your data, changing your data in your just input your data from here, and then you can run it. Okay, so for today I'm going to close here. As I told you, if you have any questions, just ask me, or you can send me email, or you can chat here. And later on tomorrow we'll discuss the correlation regressions and chi-square and a few other things. Okay, so any questions? And again, I'm telling to you again, just as I told you that it's free course, but if you're willing to donate anything, you can donate by using this two link, because all the money will goes to the for children educations. Okay, so this will I'll share in maybe later on, or maybe I'll send it to you in chat box to uh, find out the way to this. Any questions? No?
Good evening, sir.
Yes.
It is a very informative session to us, which has benefit a lot. For my enthusiasm, when I saw in some of in good impact factor journals, the authors may not publish the actual data. Means having the standard deviation, standard error, or some other ANOVA data should be, they are not been making a table in that format,
Yes,
but they will
that's why.
give the
Yes,
graphical representation itself. It isn't.
yes.
It is acceptable for the editor. Is it? I mean, editor will accept the such an journals, right?
Yes, yes, why not? You can, that is the best way to represent your data, it's graphically. But after graphically, you have to do the test, and whenever you are writing in your result, that yes, there is a significant difference between these two variety, then you have to do the t-test. Okay. So that is the way how to prepare your research paper in a way that you are represent your data in a graphical presentations, and you have to do supporting statistical test within your result. Whenever each and every statement, whatever you are writing in your result, you have to do a supporting test whether it's required. Okay, so if I telling that the production of these two variety is significantly varies, then I have to say the statistical test. Without this statistical test, that significant word does not make any sense. Okay, so that's why the statistical state is very important, and then you can present your data in graphically, no issues.
And another the factor is somebody may show that with a star symbol, star with single star, double star, and triple star
I
having.
already mentioned that one star means 0.05 means 95%, two star
Okay,
means 99%, and three star means 99.9%, 99%.
very thank you, sir. Thank you so much.
Python are both are better. Okay, it's depend upon which one is you are comfortable and which one you are learning. But nowadays both are competing to both of them are competing to each other. So Python is also good and R is also good. No issues. It's up to you which one you are using. Anything more? Okay. If not, then tomorrow's link I'll send in tomorrow email. And I told you that attendance will be tracked and then certificate will be provided based on the attendance. Okay. So yes, yes.
Uh, Sorry for the last minute question, Doctor. Thank you for your very informative and educative session. Just like to ask, are we going to receive a recorded video of this session, or we
Yes,
cannot
yes,
have?
yes. It's recorded. It's already recorded. I will
Okay, thank
upload
you so much.
in YouTube. No problem.
Okay, thank you so much, Doctor. Thank you so much.
Okay, so if there is no questions, then I'm closing the session. Tomorrow we'll see at 3 Oman time and GST+4. I mean GMT+4. Okay, so bye for now.